All right. Shall we get started?
Yeah.
Thank you so much for joining us here in person, and thank you to those who are online or will be watching this as a recorded video later on YouTube. Today we have Dr. Khaled with us. Khaled joined us this semester as an assistant professor of data science. Khaled holds a PhD in electrical and computer engineering from the University of Pittsburgh. And then he was a postdoc at the University of Pittsburgh Department of Computational and Systems Biology. The research portfolio that Khaled has built over the years is very diverse, from brain-computer interfaces and EEG signal processing to general machine vision and machine learning to modeling complex systems like biological systems or health systems. Today's talk is titled Introduction to Single-Cell RNA Sequencing Data Analysis. I'm already quite excited, and I'm looking forward to your presentation. Please welcome Dr. Syed.
Thank you for having me. Thanks for this great introduction. So are you familiar with the RNA sequencing data? All right, so basically, you know how the DNA is formed, right? The DNA, double-strand nucleic acid inside each nucleus in our cells. So everyone has different copies of the DNA, and each cell has the same DNA, right? But different cells perform different functions, right? So how does this happen? How you differentiate between a lung cell from a brain cell from a skin cell, even though all these cells have the same DNA? It's basically because of the RNA. How these cells differentiate, how these cells how they transcribe their DNA. So there is a process called DNA transcription that creates different RNA for every cell and for every cellular process, not just the cell. So every function in our body is performed because of a specific set of RNAs that get transcribed and converted into proteins, and these proteins perform different biological functions. So basically, what we mean by RNA sequencing is the process of determining the sequence of the nucleotides that form a piece of RNA. So you have a DNA, you take a copy of that DNA, and you get an RNA. So the RNA and the DNA is formed of a few nucleotides, just four. Their names are A, T, C, and G, and the different combinations create different types of proteins and different types of RNAs. So when we say RNA sequencing, we mean determining which RNA is transcribed at this time for that specific cell, and so on. So single-cell RNA sequencing is actually sequencing the RNA for every single cell. So you take a sample from a tissue, and you pick every cell, and then you study the transcription within every cell. And it's a powerful technology that allows you to examine gene expression levels, and by gene expression, I mean the copies, how which genes are copied and in what quantity, right? Okay, so as I said, it measures the concentration of the messenger RNA. It's called messenger because it moves from the nucleus to outside of the cell. It it's converted into proteins, and these proteins go to different body locations to perform specific functions. Okay, so here is the biological workflow. Basically, you take a sample from a solid tissue, and you perform dissociation to get single cells, and then you extract the RNA from these cells and perform a biological process called reverse transcription, because transcription means converting DNA into RNA. Here we do a reverse transcription to convert RNA into a complementary DNA, and then you perform polymerase chain reaction, PCR, to duplicate these DNA in order to be able to sequence them or read them. So once you have the amplified DNA, you attach some adapters, and these adapters are specific for each RNA or DNA, specific for each gene. So our goal is to quantify the number of genes per cell, right? So we need to add this adapter to tell us which gene is expressed. And then it goes to the sequencing machine, and you get a data matrix like this. It's called expression matrix, where you have rows as genes and columns as cells, right? So that's the biological workflow. We don't do it in our departments. It's usually done in School of Medicine. So here is what we do in computer science or electrical and computer engineering. This is the computational analysis workflow. So we start from here, from the count matrix or the expression matrix, and we can perform quality controls, normalization, data correction if we have different batches, if we process different samples at different times and or different locations. So you get different data batches, and you need to do a process called batch correction, and then you perform feature selection to select the highly variable genes and ignore the rest, and then we can perform clustering and cell type annotation, trajectory analysis, and differential expression. I'm gonna talk about each one of these processes right now, but this is just the overall workflow. So we start with the quality control, right? So this is what I said. Once the gene expression has been quantified, we get this matrix, the expression matrix. Each row corresponds to a gene, and each column corresponds to a single cell. And the numbers here represent the quantity, how many transcription events happen to that gene in that specific cell, right? So we need to remove poor quality cells, right? And we also need to look for the outliers with respect to the rest of the data set, rather than comparing it to quality standards. So when we perform quality control, we perform it with respect to our data set. We don't have outside or a specific quality standards. Go ahead.
What's the objective?
Yes, the objective is here. Let's go to this slide. So usually you get more than 25,000 genes in the human body, right? That's a lot of data, and many of them are not informative.
What's the end goal of this entire pipeline?
The entire
trying to model correlation.
Yeah.
From genes to trade.
Yeah. So every downstream analysis has its own goal. So clustering, you can use it to identify or differentiate between the different cell types in your sample, right? And perform differential gene expression to identify genes that are more expressed in one cell type than others. So that's one way to look at it. There's also the dynamics, how the gene expression changes over time for specific samples, right? You can also compare different conditions. So you have samples from normal patients, from normal subjects, and patients who has a specific disease, and then you compare the gene expression for subgroup of cells between the normal and disease. So you see that what happens between the two conditions.
Right. The reason I asked this is the definition of an outlier and the significance of the type of outlier you want to remove is heavily dependent on the objective.
No, here, as you're gonna see, this is performed before doing anything else, right? And by outliers, I mean something like this. Low quality cells or empty droplets usually have very few genes. So you need to remove any cell that doesn't have or at least have very few genes, right? There is also some problems like cell doublets or multiplets. So sometimes the cell before it dies, it gets bigger, and you get duplicates of the genes. So these are also cells that you don't want to include in your analysis. So you basically try to remove them from your data set. And also dying cells often exhibit extensive mitochondrial contamination. And by mitochondria, we mean, so the mitochondria is part of the human cell, and it has its own DNA. It's different from the DNA in the nucleus. So before the cell dies, you get many of these mitochondrial contamination in your expressed genes. So you need to remove all these cells before going to the lower to the downstream analysis, right? So we basically yeah, sure, yeah, no, we can have a discussion if you want.
Simply, since maybe all of the students or me or any friend don't know what is the RNA. What is the difference between DNA and RNA?
So basically, the DNA is a double strand of the nucleotides. You have ATCG, right? These are nucleic acids, and these form a single strand of the DNA of the nuclear information, right? So DNA is double strand. Each A is connected to a T, and G is connected to C, and so on. RNA is just a single strand. So what happens is that all cells have the same DNA, right? But for different functions, a specific set of genes get turned on and turned off. And by turned on, I mean transcribed or converted from the DNA into a single strand RNA, and that moves outside of the cell. So you take a copy of a specific part of the DNA, this long double strand helix, right? You take part of that, take a copy of part of the DNA, and that part represents a gene, specific gene. So when you get the RNA, that specific part, it moves to some other machinery in the cell that converts the RNA into a protein. And the protein performs a specific function, like cell growth, metabolism, whatever.
Okay. As you said right now that we have approximately 25,000 genes.
Yes.
Is that right? As a school engineer, what is the application of that gene characteristic in the information technology? What's the application?
I'm not sure if I understand your question correctly, because here we're not developing an application. We're trying to understand the biology in order to find the key characteristics of a disease development. So let's say you're studying cancer, right? And cancer is basically, sometimes cancer can happen due to mutations in some genes, right? So by studying the different amount of gene expression for between the normal and cancer patients, you can find specific genes that contribute to cancer. And then you can study their mechanism, how they change over time before the development of cancer and during the different stages.
I think that you talk still about the medical application, but when you talk about engineering, there is a couple of papers published in DNA cryptography.
Yeah.
DNA cryptography, it means that they use the characteristic of the gene. If you go a little bit back for the ASCII code, and we know that ASCII code is 8-bit, 2 to the power 8, 128.
Yeah.
And then this is only small number of combinations.
Yeah.
When you use that one for cryptography, it's weak point. But if you go with the characteristic of the gene, like you said, that 25,000, it means you have a many combinations.
No, actually, we only have 4 characters that form all these 25,000 genes. So we have we have 4 nucleic acids, the A, T, C, G, and the different combinations of these 4, you get all these complexity. So it's like Boolean.
I mean.
Yeah.
It means that whatever you have 4, but the complexity is 2 to the power, not 2 to the power, but you have 25,000.
Yeah.
It means that combination of 25,000, can we use that one for something like DNA cryptography?
I am not sure. I haven't worked on something similar to this, but I think it's possible. What I know is that some schools of engineering try to, they work on something called synthetic biology. They try to create logic gates, and they create actually bacteria that perform some logic functions. They can emit light when you put some stimulus on them, and something like that. But they try to engineer the biology, not do it otherwise.
I have a research with one of my PhD students.
Yeah.
We did a very good algorithm in cryptography and classification technique using that 25,000 gene combination.
Okay.
To create new cryptographic algorithm and new classification technique. And we published a couple of papers in that direction, and we have a patent with NASA.
Yeah.
This is the area of application of the DNA cryptography as a characteristic of that huge number of genes as a combination.
Yeah.
Because we don't want to have a limited combination as a ASCII. We use that combination and we create a new algorithm for cryptography.
Yeah, that's impressive.
Okay, sorry.
Thanks. Okay, so we basically, as I said, we filter out cells with very few genes or read counts. So we basically remove cells that are here or are below this red line. And we also filter out cells with percentage of mitochondrial reads higher than a cutoff. And that cutoff is usually 5%. So if a cell has more than 5% of mitochondrial gene, you just remove it. And also you put a threshold on the number of genes or the number of counts per cell. When I say number of counts, means number of transcripted genes, number of reads. So you basically remove these cells and these cells as well. However, you need to consider these covariates jointly instead of separately. You need to draw that figure and choose your thresholds depending on the overall picture of your data set. Go ahead.
Based on the same type of cell, or is this with a variety?
That's the variety of cells. So initially, you don't know what cells do you have. You get a sample from a solid tissue or from the blood, and then you separate the cells and analyze, count and quantify the transcribed genes. in each single cell, right? So at this point, we are still at the quality control. We do it agnostically. We don't know what cells do we have, and we don't know anything else. Just based on the number of genes and the nature of the low quality cells and dying cells, we just filter out the ones that we don't need. This is the same. So here, during this biological process, right, you get many things that can go wrong with sample collection, storage, processing, and so on. So that's why you get all of these noise. Yeah, because as I said, here you might get some dying cells, right? And dying cells have many mitochondrial DNA contamination. So you don't want to include dying cells in your analysis, right? And sometimes the counting machine sees one cell as two, and so on. So these are problems with the data collection itself. So you need to filter out all these problems. However, during quality control, you try to be as you try to be permissive as possible. And if you get bad results, you then go back and be more restrictive and so on.
How do you determine the frequency?
Sorry?
How do you determine?
It's, as I said, it's trial and error. You start with a low threshold, and then you keep changing if the results are not good.
So it'd be different for each type,
For each experiment, because as I said, now we don't know what cells do we have. I'm gonna explain how we identify cells. So after you remove and filter out the bad cells, you need to do normalization. And that's because even for the identical cells, you can get different numbers due to the variability inherent in each step. As I said, we do many steps, and sometimes you end up with different numbers for the same identical cells. So basically, in normalization, so you remember this matrix, we work on each column. So basically, you divide each value here by the sum of each column, and then multiply by 10,000. That's basically the normalization that we do in this analysis. And then you take the log, and that's called log normalization. So basically, each value here is divided by the sum of the column multiplied by 10,000, and then log transform. Then we try to extract
the column. That's going to. So sum of the column means sum the
over cells.
count of each gene, right?
No, cells.
So a column is a cell.
Yeah.
When you sum a column, you're summing two rows, right?
Yeah, because
it means that if you have two of gene number 1 and two of gene number 3, and you add them up, are you going to be able to reverse this and say that 4 is two of gene number 1 and two of gene number 3?
yeah. So here, for example, this one, you have 2, 2, 1, right? So you know that this is gene 1, gene 2, gene 3, and so on. So when you do it, you divide 2 by 5, 2 by 5, 1 by 5.
Oh, I thought you meant what you're storing is just a sum and then log. What you're
No,
normal.
you normalize first, you divide first, multiply by 10,000, and then calculate the log. So, and do you know why we take the log? So basically, these genes, as I said, we have 25,000 genes, right? Most of them are zeros. The values, the reads, for as a human, we have 25,000 genes, but when you do the experiment, we don't find gene expression for every gene. So the distribution of each gene is a binomial distribution. It's like this. So you have at zero, you have a very high bar, and then as you go on with the numbers, it goes down. So the log transformation just smooths it out, it converts it towards the normal distribution. So that's why they take the log. So for feature selection, we mean by feature selection that we want genes that are more that vary between the conditions, right? We don't want genes that have the same values over all cells. So as I said, we still need to reduce the dimensionality of our data, and we only need to keep genes that are informative of the variability in our data. So we do it very easily. We work on rows now. So basically, we calculate the mean and the variance of each gene, the mean and variance of each row, and then we get a ratio. We calculate the variance mean ratio and select the top genes as highly variable genes. Because again, now this analysis is done agnostically. We don't know what type of cells do we have. We don't know the conditions that we have, just processing on the whole matrix. So we just can pick up 1,000 to 5,000 varying genes for the downstream analysis. Then we can perform dimensionality reduction using principal component analysis. I think you're all familiar with how PCA works. So yeah, as I said, we project the high dimensional data into lower dimensional data, and then choose the highly variant dimensions to go with. And then we can visualize our data set using t-SNE. Are you familiar with t-SNE? Okay.
Just to be on the safe side, can you give a one-liner of what the TSNE does?
Yeah, actually, I have slides of how it works. So just wanted to know whether you're familiar with it or I need to go deeper. So basically, the goal is that now we have more than 1,000 genes after filtration and after removing, after performing projections. And so our goal now is to see similar cells that are similar in the high dimensional space. If we have 1,000 genes, then we have 1,000 dimensions, and we cannot visualize this. So our goal is to see gene cells that are close to each other in the high dimensional space, close to each other in the low dimensional space. And by low dimensional space, I mean two dimensional space, just to visualize it. So that's, let's assume that's the higher dimensional space, and we have clusters of cells like this. So our goal is to see them separated in the low dimensional space. So how can we do it? Do we just do a projection on one of the dimensions? Because if we do this, we may end up with something like this. If we project on the y-axis, we will get two clusters overlapped. If we do it on the x-axis, we may get another other two clusters overlap. So that doesn't help us. So in order to do this, we need to calculate similarity in the higher dimensional space and similarity in the lower dimensional space and try to match them, right? So how do we do it? So basically, for each cell in the higher dimensional space, now we are focusing on the red one. So we create a normal distribution we can first we can measure the similarity between all cells to the red one, and then we plot the similarity on a normal curve like this one, centered around the red. And then we just put the different cells on this line here, and draw a line. This line represents the similarity that we have calculated. So we will end up with something like this. Here is our red cell, the red dot, which represents the cell of interest, and then we should get similar cells close to this one, and dissimilar cells on the two ends, right? So if we create a matrix like this, adjacency matrix, we will get separate for different clusters, right? That's clear. And that's because we are calculating the similarity in the higher dimensional space, and we assume that they are separated in the higher dimensional space. However, in the lower dimensional space, they might not be similar. They might not be close to each other, right? So we need to calculate the similarity in the low dimensional space and then try to match them. So how do we do this? So basically, we start with random projection. We random project our data, and we may end up with mixed sets of points like this. And then we start with the first one again and draw a normal distribution and try to calculate the similarity or the distance from each point to the red one. And however, at this time, we don't plot a normal distribution. We plot a t-distribution. And the difference between the normal distribution and the t-distribution is basically here. So here and here. So the red one is the t-distribution. The blue one is the normal distribution. So at the far ends, the normal distribution becomes 0, which means that any multiplication will be vanished. However, in the t-distribution, we still have a value. So distant cells will still have a chance to move during the comparison that we're gonna see later. Now, we will create an adjacency matrix like this. And our goal is to move from here to here in order to match the similarities. So this matrix represents the similarity in the high dimensional space. And this one represents the similarity with the random projection. So our goal is to keep moving these orange boxes until we get something similar to this. So the algorithm of t-SNE tries to move the points one step at a time in order to cluster them correctly in the low dimensional space. So we just try moving these dots. Okay, so after that, you can use different methods. Basically, you can use Euclidean distance, right? Because of each cell. So each dot is basically a cell, right? So you can use simple Euclidean distance.
Is there some form of embedding?
I don't know. But I think for this kind of analysis, just simple Euclidean. Yeah. But I think you're right. You can do it differently by embedding the genes somehow before performing the t-SNE. Yeah. So yeah. So now you can create a figure like this. So each group of cells is clustered and separated together, because you're assuming that each group of cell is different in the high dimensional space. However, here we have actually cell annotations. We know that this group is called Clara cells, this is dividing T cell, and so on. These are different types of cells, but t-SNE doesn't tell you that the cell annotation. We're gonna see how we can annotate our cells later on. So far we've talked about quality control, normalization, feature selection, dimensionality reduction, and something else that we can do is to perform unsupervised clustering, right? And basically, the goal of clustering is to discover the natural grouping of cells. So if we assume that the specific tissue has different cell types, so that means these cell types are separated, right? So we can cluster them. Okay. Yeah. So these are three methods of clustering that we can use. However, people usually use graph-based clustering in this task. But just a reminder of the k-means clustering, you have n objects or n cells, and you need to partition them into k partitions. And the number of k is arbitrary. So you need to define k and then randomly choose k centroids for each group. You create a random centroid and calculate the similarity between each point to the centroid. And if it's close, you just assign if the distance is close or the similarity is low, then similarity is high, you assign it to that centroid, and you keep moving these centroids based on the average calculation of the samples that belong to that partition until you're satisfied with the separation, right? So you keep these iterations and stop when there is no change in clusters or you reach the maximum iterations. However, this needs you to know K, and it doesn't guarantee the optimal approaching the optimal solutions. So in order to solve this, you can repeat the process multiple times. The second approach is the hierarchical clustering. Are you familiar with hierarchical clustering? Do you know how it works? So let's skip it.
Not exactly actually. Is everyone familiar with I think it was just me that so maybe you want to.
Okay. All right. It's okay. So basically, here we try to find successive clusters using previously established clusters. So we need to establish some clusters, and then from these established clusters, we move to find the new clusters. There are two methods to do this. The simplest idea is to say, okay, all my cells belong to one cluster, and then I start dividing. The other approach is to say each cell belongs to a different cluster, and then you try to combine them. So the first approach is called agglomerative, or from bottom up. And the second approach is called divisive, where you start with all cells in one cluster and then you start to divide them. So this is how it works. So here you calculate, again, you calculate the similarity. The similarity can be based on the cosine similarity, the Euclidean distance, or correlation, or whatever. And then you group the two similar instance or two similar cells in your data set. And then you calculate the distance from each new cell to the center of that cluster, right? And you keep expanding. So now here we found out that these two cells are the most similar. So we group them. Then these two cells are the most similar. So we group them. And then this group expands because this cell is closer to the center of the first cluster, and so on. So you keep expanding until you cover all the cells. Finally, you need to decide where to cut. So here, if we cut here, we end up with two clusters. One includes all these cells on the left, and one includes the cells on the right. Or basically, it's like a cut here, right? So again, you need to define the number of clusters that you need. Okay, so that's the hierarchical clustering. Now we move to the graph based clustering. So a graph is basically a set of nodes and edges, and it could be an undirected graph or a directed graph or a weighted graph. So here in graph-based clustering, each node represents a cell, and each edge represents the similarity between the two cells, right? It could be low Euclidean distance or high positive correlation and so on. So the first approach that you can use is called k-nearest neighbor graph, right? So here for each node, right, you calculate the similarity with all the other nodes, and you connect it to k of the closest nodes or cells. So if k is 1, so you basically connect this cell to the closest one. If k is 3 or 2, you connect this one to the closest 2 or 3, and so on. However, so this approach is simple, and it's used if your data set is large and you want to do clustering very fast. However, what people usually use is the shared nearest neighbor graph algorithm. So you basically, you want to assign weight to each edge, and that weight can be calculated based on different approaches. The first one is the number, and the number of shared neighbors. So if you have a node U and a node V, so they have 3 shared neighbors, then the weight between U and V is gonna be 3. Another approach is to measure the closeness of their common nearest neighbors. And we do it by based on a very simple idea that says the closeness of 2 people is defined by their closest common friend, right? If they have a common friend that's close to each one of them, then they are basically close to each other. So for each node, U and V, right, we rank their neighbors according to the closeness to U and V, right? And then, so for example, here, if this is X1, X2, X3, then we calculate a rank between U and X1, U and X2, U and X3, and the same, we do the same for V. It's U compared to X1. And in that case, the rank is 1 because it's directly linked. So if X1 is also closest to V, then U and V should have a larger similarity or weight, right? So that's the basic idea. So how do we calculate the overall closeness of X1 to both U and V? It's basically by taking the average of the rank between U and X1 and V and X1. And so if it's 1 and then 3, then you take have that distance. So now you just basically calculate the weight as K. K is the size of the nearest neighbor list. So you have 3 neighbors. You subtract this weight, and then you assign the rest to the edge between U and V. Now, after you create your graph, you need to perform a graph cut, right? You need to decide where to cut your graph to create the different clusters. So graph cut partitions the graph into subgraphs, and each subgraph will represent your cluster. And cut size is the number of cut edges. So if we cut here, that's 2, here is 1. So the smallest cut is not always the best cut. So if you cut here, you will end up with a totally separated node, and the rest is combined in one cluster. But this one is better because it separates your data better. So how do we do it? By math. So basically, this is another, this is called a normalized cut. It avoids cutting isolated nodes and provides an unbiased measure of quality of the cut. So basically, you divide the cut S and T is basically sigma edge weights that you cut at. So here, I'm gonna show you an example. So here, if you cut here, cut ST is basically 0.1 and plus 0.2. That's the sum of the weights at the cut you decide to do, right? And volume S and volume T is basically the sum of the weights of the edges for each subgroup, S and T. So here, if you decide to cut here, you will get N equal 0.24. And if you decide to cut here, you will get 1.62. So you choose the smallest value. And that gives you better clustering, right? However, how do we know where to cut exactly? We have many options, right? So that's actually an NP hard problem, and you need an exhaustive search to do it, but it gives you unsupervised clustering, because k-means, you need to decide the number of k, and hierarchical cluster, you need to decide where to cut, but here it's automatically created for you, but it may take some time. All right, so once you created clusters, right? Yeah, sure.
You graph each node, is it representing a gene or cell?
A cell.
Or expression matrix.
The weights, sorry?
So each node is a cell.
Yeah,
So the connection, how are you getting the connection between cells?
it's by calculating the similarity, and that similarity could be Euclidean distance or cosine distance or any other kind of measurement.
Let's say that that's where the weights are coming.
No, the weights are coming from the shared nearest neighbor algorithm. So k nearest neighbor, you first, so what actually happens in reality is that you create a k nearest neighbor graph first, right? Based on the distances, right? A similarity between the cells. And then you assign weights to each edge using the shared nearest neighbor algorithm. And then you perform a graph cut.
And then are you going to call each cluster a group of cells? Is it possible for cells of the same type to have different expressions?
That might happen so under different conditions, disease conditions, you might see something like that, but it won't be as you would imagine that they are totally separated. Because let's go back to this figure here. Sorry for that. What is it? Let me go back to this one, right? So here you have red cells that are similar to the green cells, right?
Right. But I also see a lot of red cells that are all over. And then assuming that.
Yeah, because these cells, if you see the type, it's called type 2 pneumocyte, and this sample is from the lung. So it makes sense that the majority of cells are lung cells.
Right. But when you measure, let's say, Euclidean distance, and if the feature representation is essentially the same as gene transcription, shouldn't that be zero for all long cells? I think, shouldn't the distance?
No, because there are variability within each cell type. So what you're saying is that you should get this lumped together here, and
Yeah.
no, that doesn't happen in biology.
So cell type is a superclass for which we have cell subtype.
Yeah. It doesn't have to be cell subtypes. It's different, it's cells at different stages of their lives or different expression levels due to environmental factors or disease stages or whatever.
But the reason we assign the same label for all of them
Because, yeah, this is what I'm gonna show you. And at the end of this lecture, it's how do we decide that these are called type 1 pneumocyte and this is Clara cell and so on. It depends on the signature genes in each cell. So each cell has specific signature genes that give it its identity, right? So if the cell has these genes expressed, that means that cell belongs to that group, right? Regardless of the rest of the other genes. So as I said, you have
it's kind of like a chicken and egg. You have following cells that have the same signature expressions a type, and then you're saying that the type is determined by the gene expression.
yeah.
So does it have to do with functionality as well?
What do you mean?
When you say these are long cells, don't we call them long cells not just because of the signature gene, but because of what they actually end up
Yeah.
producing?
Yeah, so each cell type, so this is type 1 pneumocyte, that's type 2 pneumocyte. So they are basically pneumocytes, but they found that they perform different functionality, and that's why they separated. But
Is there an additional validation for regularization that after clustering to make sure that the cluster is actually matching the functionality of it?
so what happens in biology is that you perform some computational analysis and then you give your results to a biologist who goes and collects samples and looks at them by they experiment them in the lab. So they have to see what you're telling them happening on the real samples. So that's the validation. You cannot publish in any biological journal with just computational analysis. It has to be
The process
validated.
that you described was already based on the assumption that having the same gene expression gene signature is going to result in the same type of protein production. Is that
No,
assumption used in making the clustering more fine-grained in an adaptive feedback loop, or
it's not an assumption. So basically, you can't get two cells that are exactly the same in terms of gene expression, especially this is a low dimensional projection, right? But usually, if you take the difference between the two cells, you won't get zero exactly because some genes are different. But if the signature genes are active in these two cells, you can say that they have the same type, but they perform something at this time, they are not exactly the same. They do something different. Maybe we don't know what this functional difference
there
is.
may be functional differences, but we're still calling them the same thing.
Yeah.
Me want to, I'm
Okay.
gonna stop my questions.
Yeah.
I think we should give Khaled 5 minutes.
Okay.
And then we'll continue.
Yeah. Okay. Let me just move fast instead of going into details. So basically, that's the differential gene expression. You compare the different genes between the clusters or the different conditions. So if you collect samples from normal and disease, you want to know what happens for the same cell between the normal and disease, right? So you perform something called differential gene expression. And it's basically based on hypothesis testing, t-test between the cells and so on. But I think you're familiar with t-test. This is just in biology, we end up with performing, as we said, we have thousands of genes. So you perform thousands of comparisons, right? For the same gene between normal and disease, for the same gene, a comparison between cluster 1 and cluster 2. So you perform thousands of comparisons and you need to correct for multiple testing. So for example, if your alpha threshold is, or p-value threshold is below 5%, and you perform 1,000 comparisons, you would end up with 500 false positives, right? So you need to use methods like Ponfroni or false discovery rate to correct for this. And this is the volcano plot that we get when we perform differential gene expressions. So here is the log2 fold change, and by fold change, we mean the change from one condition to another or between one cluster to another. And this is the p-value. So genes that are spread out here are the most differentiable genes. And these are the ones that you need to care about. Then this is the algorithm that describes how we identify cells. As I said, we just pick genes that are significantly upregulated or downregulated in only one cluster. So you focus on one cluster, you see genes that are significantly upregulated with fold change greater than 0.25 and p-value less than 0.05 and expressed in more than a quarter of the cells. So, and then you take these genes as marker genes, and if they are expressed in one cell, then you consider that you give that cell its identity based on that identification. However, doing differential gene expression is not always informative, because after correction for multiple testing, you might end up with 0 differentiable genes. However, and we sometimes also, when you get these differential genes, they are not meaningful, and you cannot conclude their differences and or their functional differences between the conditions. So you want to do something called gene set analysis. And by gene set, we mean a set of genes that are known to perform a specific biological task, like cell proliferation, metabolism, or whatever. So because every biological process in our body, it happens after cascade of events. So you have a gene that activates another gene that activates another gene. So if one gene is differentially expressed, but the others are not differentially expressed, that means this pathway or this process is not as active as we thought. So instead of looking at individual genes, we look at the whole pathway and see how the whole pathway changes between one condition to another or one cluster to another. So I'm not going to describe any of the methods that we use to do this, but this is the trajectory analysis and pseudo-time analysis, which describes the evolution over time. So if you're studying biological differentiation, for example, we start with one cell, and then the cell starts to divide, and it starts to forming different organs, and each organ takes its shape. So if you want to study this differentiation process or creation process, you need time course data. You need data from day 1, day 2, day 3, and so on, and to study what happens between these times. However, in biology, not all cells progress at the same rate. So you may have cells that are at the end of the differentiation process, whereas some cells are still at the beginning of the differentiation process. So you don't want to create a graph based on the actual timing. You need to create a trajectory based on the pseudo time or the time of progress, not the time, the actual chronological time. So what we do is we do something like this. We collect the data at different times, but we rearrange the samples, rearrange the cells based on their progress or differentiation, not based on the actual time. And how do we know that? It's again based on the differential gene expression. We select specific genes that describe the progression over time, and then we rearrange the cells based on the amount of expression of these genes. If the cell has more genes that are expressed, then we know that it's at the end of the process. If the values are less are still small, then we know it's at the beginning of the process, and we can create bifurcation points like this depending on where the cells go, because they change type as we go. That's it. Yeah, if you have questions.
Thank you so much. I'm gonna abuse my position as the moderator and ask you to consider teaching a special topics course. I was always curious about most of what you described.
Yeah.
And I'm sure there is so much more there that at least
yeah, that's just scratch on the surface.
Yeah, thank you so much. I do have questions, but let's first listen to the audience questions.
Yeah, go ahead.
Just a question about cutting. So I know that during the pre-processing, you didn't specifically go to find out the labels of the cells. So they were just a general mix. Whatever you can, you don't know what the process is. Was that at all in terms of the balance? Is that how the graph gets cut? Because the measure I saw there was a bias, but I'm not sure whether or not it had any points or how what that at all would actually affect the types of excitement.
Which variable is that?
So
Yeah,
with that unbiased.
yeah, yeah, the normalized cut. I don't think so, because it's, if you look at the way that we create the initial graph, the unweighted graph without weights, it's based on the similarity between the cells, right? Regardless of the clusters of the actual cell types. So
Because the cut, right, was
yeah.
But if there's a whole lot more, say, other classes that makes it imbalanced, even though.
Yeah, you mean the number of cells. If the number of cells of specific cell type is greater than the number of cells of other cell type, right? That would give us an imbalanced graph. And so this is actually, if you go back to the methods, our initial goal is to find the weak connections between the two graphs, between the sections of the graph, because a weak connection means a separation, right? Exactly. And the minimum value means the weak points. So here, if you cut here, you are cutting 4 connections. Here you're cutting 2. And these weights also is calculated based on the common shared nodes. So if you have imbalanced data set, right, you would expect that the cluster with the small samples to be not connected to as many cells as the cluster with many cells, right? So you would find a weak connection between the two. And it should be, yeah, I think, yeah, go ahead.
Is there a way to reduce this problem to an iterative mean concept?
I think so.
It's still NP.
Yeah.
But
So there is
curious if for weighted graphs, This can essentially become something like updates over the minimum cut, where you also want to balance weights or average weights in each cut. Is that right? Is that how you formulate it?
I think so, because here we are looking the minimum weight. So if we cut at the minimum weights, that's the best that we can do. But yeah, I'm not sure actually.
But
I have a single question.
Another question, what's the reason behind that we need to evaluate within a cell, the cluster by itself? What's the reason behind it?
Because it's not gonna be informative. Yeah, but I think biologists have discovered all kinds of cells so far. It's rare to find a new cell type, so, right? We're not classifying. We're not classifying. We're labeling. Right? We're not looking for new cell types in nature. We're labeling the data set that we have. So again, we have a mixed set of cells, right? When we perform an experiment, we get tissue samples and we separate the cells, but we don't know their identities yet. And then we cluster them in order to separate each group of cells, right? It's very it's rare to just get one cell from a specific cell type. You usually get a group of cells from a tissue. Yeah, this is what I said at the beginning, is that usually you're not going to find new cell types because biologists have found almost every cell type in our bodies.
you don't assume anything about our knowledge of the cell types in this approach.
Yes,
So you can't find new cell types, but you just don't know and don't care. It's like that.
yeah, but his question is about finding one cell that could be very unique, and it could be very informative that you have found something new. No one ever discovered it, right?
Yes. If the same cell we are examining at the part with body organs, we end up with this cell, or I don't know, like a very specific state of
So, for example, you can say that this cell could be in one cluster, right? And then we say that this one is very interesting. Let's take a look at this one specifically. What I'm saying is that that's unlikely to happen because it's very difficult to find rare cells with these simple approaches or just computational approaches. You need to see some phenotype or something that's happening in the biological system that you cannot explain, and then you go and investigate the role of these cells. But with this computation, as I said, you make so many assumptions. You can't be confident that this cell is something unique. It's basically The simplest assumption is that this cell belongs to the closest cluster.
That's actually a beautiful question, and a beautiful point. It's rare to find an individual unique cell that is important because we are explicitly filtering them out. Isn't that right? In the outlier detection, in the thresholding, we are trying to get rid of things that aren't similar to other things.
But in terms of the number of genes and number of count reads, which could reflect errors in the data collection and data processing.
It's essentially banking a lot on the law of large numbers to overcome the gap between a steady state and dynamic.
Yeah, exactly.
And is that banking, is that investment a wise investment in a dynamic system? Why does that work?
So it's not just banking on the rule of large numbers. It's banking of the nature of the problem, the nature of the biology that you have, because when you filtered out these cells, you know that these are dying cells or these are bad samples, right? And what you end up with is a good quality cell. What you're saying is that rare cells will be similar to dying cells or the cells that we filtered out, which is not true. It could be, but usually it's not, because this is an assumption that rare cells in our body will behave like cells that should be filtered out. What I'm saying is that if we found or if we find a rare cell type, it should act like normal cells, but it should be in a different location in the
In a different
cluster.
If it's among the top 5,000, 6,000 variants to mean rank.
Sorry?
So you rank the
varying genes.
expressions
Yeah.
based on variants.
Yeah.
If you have a very rare cell, is it going to make it to the top 5,000?
No, but this task is not about looking for the rare cells.
Yes, that
Right.
is about
That's not causal modeling.
you're looking at relationships between genes. So far, it's just cell identification. But the cell identification is going to be used for the causal.
It doesn't have to be that way, because
When it comes to disease identification, isn't the task there to see what expressions of genes, in other words, what cell types are involved in the emergence of disease?
not cell types. You're looking for genes within cell type. So these samples, these are the same cell type, right? But you have the same group of cells for patients and for normal subjects. And then you compare the pneumocytes between normal and disease. So you're looking for the differential gene expression for a specific group of cells between two conditions.
Why?
Why not?
Why are we? What does that tell?
It tells you what happens for these specific cells between normal and the cancer, for example. Because as you said, these are they are not the same. There is variability, and this variability could be due to a disease or external factor or whatever. That's not causal. That's just
discrimination. You are discrimination in statistical. So you are trying to discriminate between cell types or gene expressions, according to relevance with disease. Is that right?
Not relevance, it's
Correlation.
correlation. It's basically correlation.
What do you do with that?
So what you do is that if you find that a gene, a specific gene, is highly expressed in cancer patients, for let's say you have epithelial cells, for example, and you find that that gene is high in cancer and low in normal, right? So that could be a target gene for therapies, right? If you suppress its expression, you can move from cancer to normal.
So, do you see why I use the term causal?
It's,
You see a correlation between the expression of gene and cancer. And you say, so if we target one variable, the other variable is going to be affected.
again, that's not causal, that's an assumption of causality, right? And that's not what you should depend on finally. But so in biology, you basically do trial and error. You find a correlation, you try to find a causal relationship, but sometimes you don't. But still, if you see a strong correlation, you start administrating drugs. And if you see change, then you say, we did that experiment, we inserted that drug, and we saw this improvement in outcome, but with confidence interval. But again, it could be a coincidence. And after 10 years, some people come and say this drug is not the actual cause of
reduction.
exactly.
But in a lot of cases, it doesn't happen.
Which cases?
And in a lot of cases, the assumption that the correlation is causation works out. That's why we have a pharmaceutical industry, right?
Yeah.
Why does that work?
Because we don't understand the complexity of the biological system. This is the best we can do so far.
You're right. The system is complex, and our understanding of complexity and this type of modeling tells us that this shouldn't work so many times, but it does.
So that's not modeling, actually. That's just descriptive analysis.
Descriptive analysis based on modeling assumptions. I still call that modeling. One
Okay.
of the modeling assumptions that you made here was a steady state description. You assume that the samples that you take are descriptive of the type of system that you're trying to identify.
At the time point of the data collection. So it's a snapshot in time. It's not assumption about steady state or about the transient response. It's just assumption about the time being, the time point that we collected this data.
I suspect that somewhere in the last few slides that you kind of skipped, you do talk about the transient nature.
That's during the pseudo time analysis. Now, in the pseudo time, we have data from at different time points. So at the beginning, we have data at a single time point, right? But if you want to perform dynamic analysis, you need to collect data at different time points. But that doesn't mean that we assumed a steady state at the beginning. It's just a snapshot in time. We perform the analysis. We come up with some descriptive analysis that could be different if someone else collects data, right?
I
So
don't want to bore others. This is
yeah,
something that-
biologists don't think of this problem as a dynamic system from a system point of view, right? They just, as you said, it's based on correlation. You collect data, you see changes in between different cases, and then you make an assumption. And if all people perform the same experiment and see the same changes in genes, then we might start a clinical trial. So a clinical trial doesn't start unless you have so much evidence about that specific correlation. So
I don't want to bore others. I'm just gonna probably use this from in 5 minutes.
okay.
But I know there are a lot of people who still want to ask questions. Did you have a question?
Can you go back to the slides, which is the 6 cell or 6 gene?
Um,
6 node or 6 cell or 6 gene connected with each other.
this
When you
graph,
said top, yes.
yes.
Okay, my question here, the red and the blue spot here, this is cell,
Yeah, these are cells.
okay. It means that there is interrelation between the cell with each other.
So a connection means there is similarity. They are close to each other based on the Euclidean distance or the cosine similarity. Because so each cell, let's say, has 100 genes, right? And you have different value for each gene. So you take the difference between 100 value and 100 value and calculate the square of each difference and take it under the root.
What does it mean by 0.1, 0.3?
That's the weight of the graph. It's calculated using the shared nearest neighbor algorithm. That
I mean physically.
physically, it means that these two cells are not close to each other. So the shared nearest neighbor assigns a higher weight if the two cells are close to the same group of cells. So if one cell is similar to 3 cells and the other cell is similar to the same 3, so that means they are close to each other because their friends are the same.
Be that 2.1, 2.3, it means that this cell and this cell near from each other. and
No,
this
they
cell
are far. The less the value is, the far they are from each other.
are at a
They're
distance point.
not
Yeah,
this,
yeah,
but they don't have the same friends.
I mean,
Yeah,
near as a-
yeah, exactly.
Not specifically, but
Yeah.
so you and Khaled are similar to each other in terms of place of birth, or you're both from Egypt.
Who
Bahar,
said that we are?
I'm going to make that assumption. That's the hypothesis. And Bahar and Arshad are from Iran. You two are close, similar to each other, close to each other. You two are close to each other with respect to the descriptive characteristics.
Yeah, that's fine.
So the
My question is
closeness,
0.1, 0.3. This is built on that.
so
Yeah,
yes,
yeah.
this is, you come up with a number of descriptors. You call them features to represent each cell. In here, the features are gene expression.
Yeah,
Is that right?
yeah, exactly.
And
And
then
also
we,
from that distribution here, you have one cell connected to 2 cells and one cell connected to 3 cells.
Yeah, that's actually a visualization of the problem. These are not actual cells. This is just a toy example that describes the problem.
Okay.
Right?
Because my question is always go to my background as a network.
Yeah.
How the
Yeah.
network connectivity-
No, yeah, that's just a toy example.
Okay. Thank you so much.
You're welcome.
so, just to be on the safe side from a rules kind of perspective, let's thank you again and let's
You're welcome.
have the official Q&A. If we can, please stop the Zoom recording
Okay.
and
Okay, let me see.
so I really want this to go on, but do you all agree that Khaled should teach this?
Yes.
Next semester, Khaled, if you teach it, I'm gonna